So thank you very much, and thank you, Felix, for the introduction. My name is Elisa Riedo. I'm a professor at the New York University in New York, and the title of my seminar today is Thermochemical Scanning Probe Lithography, tc-SPL. So the basic idea of thermochemical SPL is similarly to thermal SPL, the use of a hot probe, a hot tip. But the concept here is slightly different because what thermochemical SPL is trying to do is actually to activate by using heat a chemical reaction. And as I will present later today, this can give rise to grayscale patterning of chemistry and functionality at the nanometer scale, with simultaneously also the possibility to do a 3D topography patterning. The concept of thermochemical nanolithography was actually demonstrated in 2007 when I was a professor at Georgia Tech. In fact, before moving to NYU, I worked at Georgia Tech. I was a faculty there for 13 years. And a lot of my work on thermochemical SPL was actually developed when I was at Georgia Tech in collaboration with Bill King, who at the time was also a professor at Georgia Tech and came from IBM, where he did his postdoc. So in fact, Bill King worked with Binning and Vettiger at IBM in Zurich, moved to Georgia Tech, came with the technology of hot tips and thermal probes, and talked to me about what can we do with this amazing technology. And that was when myself and my group, we started to think about using the thermal probes for activating chemical reaction, and in this way to actually develop a method for nanopatterning surfaces. Later on, IBM worked more focusedly on the thermal SPL in the sense of, as you have seen in many other seminars, right, using the heat for the evaporation of a resist, PPA, which is excellent for the topography patterning. And in 2013, Swiss Litho, the company, the startup company, came to life thanks to Felix, and let's say the rest is history. Okay, so let's see a little bit what you can do with this concept of thermochemical SPL. I must say that sometimes myself as well use tCSPL, just because now everything is becoming so broad that sometimes it doesn't really make sense to keep two names. So tSPL and tc-SPL may be actually used interchangeable, but let's try to stay focused for this seminar on the idea of the chemical reaction, of the thermally activated chemical reaction. So, so far in my group, what we have demonstrated is the possibility to use a polymer and then activate the chemical reaction, expose some functional groups, and then use these functional groups to actually attach other molecules like proteins or enzyme, and also to create chemical gradients of these biomolecules. Next to that, we also have developed a few methods for using this thermochemical approach for 3D patterning, so for topography patterning of the polymer. We also use thermal and thermochemical lithography for building, and I must say for fabrication of electrodes. In this case, I would say is mainly thermal SPL, not thermochemical, so using the nanofreezer technology with PPA. And Shaori Wang Zheng showed this last week, I believe. And you can also use the thermochemical lithography for magnetic patterning. And finally, you can use it for local crystallization. So you can use a salt gel of a ceramic material, and the heat can be used for the local crystallization, for example, of ceramic nanowires like PZT, so ferroelectric and piezoelectric ceramic nanowires. Or going back here in the electronic materials, you can use it for reduction, for example. So you have, for example, graphene oxide, and by using heat, you can reduce graphene oxide back to graphene. So again, is a thermochemical reaction locally activated by the hot probe. Okay, so today we're going to focus mainly on chemical patterning with ad hoc polymers. So similar to what you have seen for tCSPL, you need a good polymer, a good resist that is thermally sensitive. If you want to do a chemical patterning, what you need is a polymer that not only is thermally sensitive, like it evaporates, like in PPA, but you want something slightly different. You want something that changes the chemistry due to the presence of heat. So this is a polymethacrylate copolymer, and it has some protective groups here, which makes the polymer inert and relatively hydrophobic as well. However, these groups can be thermally deprotected by applying a temperature of about 125 degrees Celsius. So what you can do, you can heat locally the polymer, and you can deprotect the polymer from these groups, and you can expose carboxylic acid groups. So this is what you have here. So this, the white area here is where the protective group has been removed, and it is where you have carboxylic acid groups. This is a friction image. Why friction? Because friction tells you that some chemistry is happening here. So next step was, okay, can we actually think about creating patterns of carboxylic acid groups or even of amine groups, which are actually perfect functional groups to attach biomolecules, and then deep in the solution of the biomolecule of interest, say proteins, and use some bioconjugation scheme to attach the biomolecules here selectively to the patterns of amine, then some wash protocol, and generate a pattern of proteins, for example. So that was the idea. This is the second type of copolymer. So this is a copolymer where always a methacrylate copolymer, where the functional groups now that are exposed after heating are actually amine groups. Now, you follow the same protocols, and you can also think about different bioconjugation steps. So, for example, you can use deep in a solution with SPDP and transform your amine groups into thiols group through a second washing with DTT. Or you can bioconjugate the thiols group with some NHS biotin, and then have a pattern where you have biotin, sorry, here, the red one is biotin, you have thiols here, and you have amine here. So you have 3 different functional groups. And then use these 3 different functional groups in a solution with proteins, for example, which can be bioconjugated specifically in each type of functional group. So on thiols, or on amine groups, or on biotin. And these are the results. So these are different type of proteins that have been bioconjugated to the amine patterns, and the different colors shows the different proteins. And this is instead is DNA, so showing that you can also pattern DNA. Okay, next step. Next step is the idea of a chemical gradient. So can we create a chemical gradient? And the answer is yes. How do we do this? Well, the deprotection of the thermally protective group is a chemical reaction, right? So it is actually a first order chemical reaction that follows an Arrhenius law. So we can write the rate of the reaction with a attempt frequency, the temperature that we're using, and the activation energy of the chemical reaction. Then we can write for this type of chemical reaction, we can write the equation of how the probability of the reaction change with time. And it follows a first order chemical reaction equation, and then with K being the rate of the reaction. So when you put the two equations together, you can see that by varying the temperature, T, or by varying the variable of time. What is time? Time is the dwelling time, meaning the time that your tip stay in contact with the surface. So the longer you stay in contact, the higher the temperature, the more probable is that the chemical reaction happens. The faster you move, less time you stay in contact, less probable is the chemical reaction. So everything is inside here, so it can be very nicely analytically predicted. And so you can really build a system of an analytical equation that predicts by knowing the time, so or the speed, so the dwell time, or the speed, which is essentially the same thing, and the temperature of the probe predicts exactly what is the probability that your reaction happens, which means gives the probability of, gives you the probabilistic amount of how many amine groups you have exposed. And so you can build complex pattern based on this. So you can have an input image, in this case was a Mona Lisa image, and then you can use your equation and know in each point how fast or how high the temperature should be in order to expose a certain amount of amine groups. And once you have that pattern built, you can dip again your sample in a solution, bioconjugate the amine groups with, for example, dye molecules or with fluorescent proteins. And then you go with your sample in an optical microscope, in a fluorescence optical microscope, and this is what you get. A fluorescence image of the Mona Lisa, where you see where the amount of amine is given by the fluorescence intensity. Here are other images. This is a Adams rose picture, and in this case, instead, this is a laminin. So this is a protein used for guide neurons grow. This is a gradient of laminin on the surface. The resolution is sub 100 nanometer resolution for this type of gradients. So these are the smallest gradients, chemical gradients built so far. Other steps here are, can we now couple and have simultaneously control of chemical patterning and 3D topographical patterning like we have seen with PPA. So these are some example. This is the same copolymer This is done with the nanofraser, so it shows using this copolymer, not PPA. So it shows how you can pattern the topography. The mechanism here is a little bit different. It's not the quick evaporation, but it's really more the deformation of the pattern due to the local heat. So it's more a deformation-driven mechanism. There is also evaporation of the protective group. So it's a combination of the two. And this is the chemical image. So it shows that where you have heated, so you see this is deeper, right? The topography is indented. So you have heated more here. Well, there are also more amine groups here. So the fluorescence is higher. In this case, it's a very different type of thermochemical reaction. This is a precursor polymer of PPV. PPV is a conjugated semiconductor polymer, which once the precursor is transformed in PPV, PPV is also fluorescent. It's semiconductor and fluorescent. So what it shows here is that the image is showing some fluorescence, but it shows also a 3D patterning, because when the precursor is transforming PPV, there is also a shrinking process. So you can also change the topography by looking at the, sorry, not a shrinking, a swelling process. Okay, next step. Okay, what is the ultimate resolution, chemical resolution that we can achieve with this method? And can we really combine this with an extreme high topographical resolution? Why we want this? So this was and still is the vision for why we started to go into this direction. So we wanted to add another dimension in our patterning, right? So we are patterning the chemistry, one. We are patterning the topography in 3 dimensions, X, Y, and Z. So we have 4 dimensions of patterning, of control. Now we are adding a 5th dimension, which is time. Can we create patterns that change with time? So they form and then dissolve. So dynamic nanopatterning or transient nanopatterning. How do we do this? Well, we can combine the chemical patterning with biocatalytic self-assembly. And this self-assembly, I just saw the time, 39 minutes. Felix, I'm okay. Can I keep going?
Sure, go ahead.
Good. I saw 39 minutes, so I saw, okay, so I will try to go a little bit faster. So you
just talk for maybe 20 minutes maximum.
Oh, good. So I don't know why I have 40 minutes here. Good, thank you. So, so what is biocatalytic self-assembly? So essentially, this is a reaction that is triggered by enzyme. And once a peptide solution, certain peptides are embedded in a solution in presence of particular enzymes, what you have is a self-assembly of the peptides to form this very interesting fiber structure. And not only these fibers can form, but you can also use a biofuel to stop the reaction and then to reactivate the reaction. So with an external chemical trigger. So the idea here is to pattern enzyme on a surface with tc-SPL, where ideally we can have 1 enzyme per spot, and then bring in a solution with these peptides, the biofuel, and start the growth of the fibers in each spot. And if we can also pattern different enzymes, we will have also different types of fibers. So what it shows here, it shows that depending on the biofuel, we can activate the fibers green and blue, or we can activate the fiber orange, green, and blue, or only the fiber orange. So we can control in time what nanostructures form. So this is the general idea. Our part, so this was a collaboration with Professor Ulein at CUNY, and our part, of course, is can we create patterns with single enzyme on the surface. So this work was performed in my lab with my student Shang Liu, and we call it Shun, with this type of scheme. So we start again with the pristine polymer that I showed you before. So this is a methacrylate copolymer, which expose amine groups, okay? Then, as I say before, you can either expose amine groups at a certain temperature, then dip in an enzyme solution, or you can also, we realized, if heating at much higher temperature, you can go through a different chemical path and passivate the polymer. So depending on the temperature, you can change the amount of amine exposed. You can decide to passivate the polymer, or by controlling, and this is by controlling the temperature, okay? Like I say before. So the gradient, right? The temperature can control the density of how many amine groups we can expose. However, by controlling the load, so how much what pressure we apply between the tip and the surface, we can also control the depth of the pocket where the enzyme we're going to stick. So the idea here is to be able to control the density of the enzyme and the 3D topography of the enzyme. Then the bioconjugation that is used here is an electrostatic bioconjugation. So what we're going to do is we are going to attach the enzyme by using a sulfonated group which are negatively charged. So these sulfonated groups are conjugated to the enzyme, and they're negatively attached, negatively charged, and so they are strongly attracted by the NH3 plus group on the surface. Remember, all this is happening in water. This reaction is happening in water and at room temperature. So first you create the amine, and then the reaction of this electrostatic attachment happens in water. So it's a very strong, actually, it's a very robust, even if it's electrostatic, it's very robust. This is a slide showing the surface passivation scheme. So here what you have is the pristine polymer, and as you can see here, unfortunately, the pristine polymer, even if it is pristine, when you dip in the solution with the enzyme, some of the enzyme will still attach to the polymer. So you are not able to, remember, this enzyme are bringing a very strong negative charge. So somehow the polymer is still attracting this enzyme. So the non-specific binding is quite elevated. However, if we heat at very high temperature this area, we can actually expose, as you can see here, some COOH groups, which are negatively charged. And so these carboxylic acid groups are actually repulsing deposit the negative enzyme groups, and so they don't want to stick to the passivated area. So you see how dark is now the passivated area. On the other hand, the inner square is where we have the amine groups, so the positive NH3 groups are instead attracting and quite strongly keeping attracted the negatively charged enzyme groups. So this is the scheme, and this is FTIR to confirm the presence of the different functional group that we are exposing. Okay, this slide shows instead how we can control the enzyme density with the temperature, so the percentage of amine groups, and the pocket depth, meaning the 3D, right? How deep is the indent that we are making. So we can calibrate, so we can change temperature and load. This is showing the density of the amine groups. This is the fluorescence showing how it changed with the temperature. This is instead is a calibration where we change temperature and load at the same time. So you have the different curve here are indentation versus load for different temperature. And this is the two main equations that we are using. So we are using a modified Hertz model, modified because we assume that the Young modulus of the polymer becomes lower and lower with increasing temperature. So we can quite well control the pocket depth by increasing the temperature, because the softer is the polymer, right, the more for the same pressure we can make a bigger indent. This is what this equation is telling us. And with the load, so the higher the pressure, the bigger the indent. So this equation is essentially giving us the fitting for the experimental curves, and together with this equation, which is similar to what we have before, but now we have also to keep in mind that there is a passivation going on, so the activation of negatively and positively charged molecules, and that's why here you have the fluorescence have a maximum. So in the beginning you expose only the positive charges, so the enzyme won't attach. But then high temperature you start to expose also the negative charges, which repels the enzyme. So you have a maximum and then a decreasing. So by playing with this temperature that control the density and the temperature and load here that control the depth, you can precisely know how much enzyme you have and how deep your pocket it is. So you have a extreme good control of the two parameters, depth and density. Okay, so these are other images showing that, you know, you can do AFM to confirm the fact that first you put the amine, so you see there is this indentation, so formation of the pocket, so the indent, or let's say this is the pocket, right? This is what we are doing. We are creating a pocket, and we then put the enzyme on it. So this is with enzyme, so after dipping in the solution with the enzyme, and you see that the indentation disappears because the whole pocket now is filled with the enzyme. And you can see this at different scale. This is a 100 nanometer pocket filled with enzyme. And this is the corresponding fluorescence demonstrated the presence also of the enzyme. Then we pushed the resolution. And what you can see here is that we are able actually to make pockets where the size of the pocket is 8 nanometer. The size of a single enzyme is about 6 nanometer. So these are single enzyme pockets. And these are nanowires where each wire actually has only one enzyme in it in terms of width. So our single enzyme nanowires. And this is the demonstration by fluorescence that indeed these wires have been filled with enzyme, and you can see also in AFM here. Then the last goal was, can we create single enzyme patterning over large scale? So what you see here is the logo of NYU Tandon. So Tandon is the engineering school of NYU where I work. So every letter here, everything that you see here is made of single enzyme lines. So the idea here is we built this N, again you see the nanofraser image. So this N, you see, zoom in here, is made of lines, and each line is about 8-10 nanometer width. Then you dip in the solution with the enzyme. The enzyme stick inside each single enzyme line, and this, then you go in a fluorescence microscope and you demonstrate that indeed your enzyme are filling your single enzyme lines. And the scale is pretty large. So this is 100 micrometer. Yeah, we were quite happy about this result of large scale with single enzyme resolution. Okay, so what next? So right now we're working with Dr. Giuseppe De Peppo at the New York Stem Cell Foundation on biomedical application. What type of biomedical application? Well, right now we are focusing on tissue engineering. So can we actually design tissue microenvironment with the morphological and physiological features resembling those in vivo? Meaning, can we do a precise chemical and topographical replica of the tissue microenvironment? So there are a lot of attempts around in literature, right, where the tissue has been where the interaction between cells and micro pattern or nano pattern have been studied, and some tissue engineering study, a lot of actually of tissue engineering studies have been done, but mainly either the scientists used self-assembly, so quite a disordered type of microenvironment fabrication, or they use conventional fabrication methods like optical lithography or E-beam lithography, where very, how to say, very ordered and simple structure can be fabricated, like pillars. So the typical example is an array of pillars. But you can see how a tissue, a human tissue, is very different from an array of pillars, right? So the capability of tc-SPL and tSPL would allow us to reproduce the complexity of the human tissue in 3D, and maybe even the chemistry. So this is the idea. This is a SEM image of the bone tissue at the nanoscale, is showing this microfibrils, and the size of this microfibrils is really accessible by tc-SPL. So you can see that it's around 200 nanometer, the larger the distance, so the diameter of the fibrils is 200 nanometer, and then you have all these tiny nanoscale features that have size between 60 nanometer, and the height here is just 20 nanometer. So these are all scales that we can actually assess with tc-SPL. So this is a preliminary data where we use the nanofraser to replicate the bone tissue image. So this is an in vitro bone tissue image, and this is the perfect exact replica of it with the nanofraser. So this work has been submitted for publication, and as I said, this is a collaboration between my group and the PEPO group at the New York Stem Cell Foundation, and Alessandra Zanut, who is a postdoc in my group, and Shen Liu, who is a PhD in my group. So one important thing since the work is only submitted, I cannot give you too many details. Don't want to, otherwise my collaborators are going to kill me. So I cannot give you too many information. You will have to wait until the paper is published. However, the key thing here is that the polymer that we use is again the methacrylate copolymer, which means we not only can control the topography, but also the chemistry. So we will really have access, right, to this complex physiology and topography and morphology of the human tissue. Okay, to conclude, I switch gear completely and move towards electronic devices. So Ray Cheng already talked about contacts on molybdenum disulfide for field effect transistor fabricated by thermal lithography, and how these contacts have superior performances compared to conventional methods like e-beam lithography. So I'm not going to go too much into details of this, just for the ones of you who were not present at Ray seminar. Let me tell you, the idea is that you use a two polymer stack on top of a monolayer of molybdenum disulfide deposited on silicon oxide, PMGI and PPA. So you use tCSPL to evaporate locally PPA, then you use some chemical etching for the undercut here, and then metal deposition, and finally lift off. With that, you can pattern complex design of electrodes on molybdenum disulfide. You can create also top-gated field effect transistor, where the top gate, you can use the nanofraser beautifully to really image where the two electrodes, source and drain, are, so that you can visually see where your top gate, you see, in the middle can actually be fabricated. So you don't need at all markers. So that's a big advantage compared to e-beam lithography. Other advantage from EBL is the cleanness of the surface, as you can see here. Okay, so based on this, we went further and we said, okay, now not only we can fabricate devices, can we also dope using the idea of thermochemical reaction? And this is what we have done. We have built a gas flow chamber. So this work was done in collaboration with Ray Cheng in my group and Annalisa Calò, who was also a postdoc in my group. She's now in Barcelona. And as you can see here, there is a gas flow chamber where you can not only heat, but you can expose your surface to certain gases. So there is a chemical reaction between the heat and the gases present in the chamber. And so depending on the gases that you choose, you can achieve either N-doping or P-doping. So for P-doping, for example, you use a mixture of HCl and water, and for N-doping, you heat in nitrogen. So you can control N and P doping, so you can have a bipolar doping, and you can then fabricate a PN junction. This is what we have done. Again, the electrodes have been fully fully deposited with tCSPL. So the whole device from doping to electrode fabrication has been completely fabricated with thermal and thermochemical lithography. And this is the rectification ratio, 10 to the power 4 of the diode, which is really an extremely good result comparable with state-of-the-art PN junction in molybdenum disulfide. With that, I finish my seminar, and I would like to thank people in my group. I already mentioned Xiaori Zheng, Shangyu Liu, Annalisa Calò, Edoardo Albisetti, Alessandra Zanut, and who are the main people working on TC and tSPL. And actually, also they were working on tCSPL, but these are people who left already my group. And Francesco, he's still in my group, and Li Wenxie and Xin Liuzhu are also still in my group, but worked a little bit less on this project. And then my collaborators at NYU, Sharjerdi Davoud for the electronic measurements, Ulain at CUNY, Marcus Vack, who actually developed the methacrylate copolymer for all the experiments that you have seen, Bill King for our initial collaboration, and also Jennifer Curtis for our initial collaboration, and then all the team at Swiss Litho, which is now, sorry, Heidelberg Instruments, who have been fantastic and always willing to help, and also actually a great friendship developed, and the funding agency, of course. So thank you very much.